So thank you. And in the next 10 minutes, we'll explore the recent history of genomics in monitoring and improving animal genetic resources from microsatellite to advanced sequence technology. And just a note for animal genetic resources are a cornerstone of global food security, providing us with diverse breeds adapted to meet human nutritional needs. And they're not only vital for sustaining livelihood, but also hold a deep cultural and economic significance in many communities. And their diversity is an invaluable resource for adapting to changes in production environment or climate. These resources, of course, face significant challenges, including loss of genetic diversity, economic sustainability, and environmental impacts. In both senses, they are increasingly vulnerable to the effect of climate change. And at the same time, they contributing to environmental pressure, especially with greenhouse emission. Additionally, animal welfare is a growing concern that must be addressed to ensure ethical and sustainable practices in managing these resources. Genomics played a major role in understanding genetic diversity, which is pivotal in livestock management and improvement. And this is because it provides a foundation for guiding conservation efforts, ensuring we preserve also rare alleles. They may hold a key for future adaptability and resilience. By assessing genetic diversity, we can mitigate the risk of inbreeding, which is often resulting in reduced productivity and for sure health issues. This understanding also shed light on molecular mechanisms that influence traits, allowing us to make more informed decisions within breeding programs. Additionally, improving estimates of breeding value through genetic tools enables precision in balancing selection for the desirable trait and the need of conservation. I think Jack will talk about that later. And finally, the enhancement of this breeding program not only increase productivity, but ensures sustainability for the future. So here is a timeline of advances in the last 30 years. Were marked by significant milestone in general like agrobiotechnology, including very strong breakthrough like cloning, genome editing, advanced genomic data application. These innovations have brought a revolution in the way we monitor and improve genetic resources. Over the years, we have transitioned, and you see in the bottom of the slides, from microsatellites in the 90s and early 2000s to widespread adoption of single nucleotide polymorphism. We'll see these markers and advanced sequencing technology later and till today. Advances in the early 90s, advances in PCR technology enabled easy and cost-effective amplification of microsatellite region. Microsatellite markers are a short, repetitive DNA sequence, usually 1 to 6 pairs, making them a very popular tool for genetic studies. In livestock, researchers started using microsatellite to analyze genetic diversity, population structure, and parentage. And FAO supported the development of microsatellite panels for standardized assessment of genetic diversity in domesticated in farm animals. The advantage of this kind of markers were mainly represented by the fact that they are highly polymorphic, enabling detection of fine-scale variation. They were affordable compared to other molecular markers. They use simple protocols, make them doable, accessible to a wide range of research. And they really proved useful for estimating genetic structure and were applied to all the major international effort in evaluating the origin, the domestication, the genetic diversity, both within and across a very large number of breeds and population in all the livestock species. Really, like tens and tens, like hundreds of papers came out on using these markers. The major disadvantages of microsatellite markers is definitely the limited genome coverage. They are not spread, across all the genome. Moreover, they are labor intensive and time consuming. I'm thinking some of you may have spent a lot of hours into that. And microsatellite results can be difficult to replicate across laboratories. Also, they do have a high mutation rate and very limited scalability. So all these factors are limiting future potential, although they're still in use in many species. With the year 2000s, research institutions around the world joined forces to produce reference sequences for many livestock species. And reference sequences are the backbones, the basis for identification of millions of single nucleotide polymorphisms, so single mutation that differentiate individuals and populations. And SNPs, have become available in large number along the genome of many species, and they offer greater genome-wide coverage, enabling more comprehensive studies of genetic diversity and genome association study with traits of interest. The ability to pinpoint single base changes increases the accuracy of diversity estimates and improve the identification of fine-scale genetic structure. The standardization of these markers ensure that they are very reproducible and comparable, and this is critical when operating on larger scale collaborative studies. They're scalable and cost efficient, and today there is a very extensive bioinformatics support for SNP analysis, which enables robust statistical modeling and bioinformatics. And also they have a low mutation rate, so this stability ensures the reliability of studies, also genetic history and all the population dynamics from the past to the present times. So many SNP chips, what we call these SNP panels, from different projects and provided by different companies, ranging gene size from 10,000 to 1 million SNP. They have become available and affordable for farm animals, alongside with whole genome resequencing technology, so sequencing of single individuals also. And when choosing between these two technology, the choice should be guided by the specific species we're analyzing, and the goals of the research or the application of the research. So whole genome sequencing is the gold standard for comprehensive unbiased genetic analysis, and in particularly is useful for novel or underrepresented or very diverse populations. However, of course, it has high cost and very high computational demands, and this makes it less accessible for really large-scale studies. SNP chips, on the other hand, they are cost-effective and scalable, But they're limited what is called ascertainment bias, which is, basically they are derived from a set of population, and they may be not applicable to novel population. So for optimal outcomes, a combined approach may be used, like a SNP for large-scale routine monitoring and whole genome sequencing for detailed study or when working with novel populations. From short-read sequencing, enabling SNP discovery to these new long-read technologies like Oxford Nanopore, these sequencing advantages in technology of the sequencing significantly enhance the resolution and the accuracy of genome studies. And this is also largely applied in livestock breeding programs, including programs for selection and conservation efforts that have attained a very important success due to the affordable genomic prediction, particularly in dairy cattle, I would say. Well, novel traits, and we see in these slides, are explored. In this case, selection and understanding the traits of to mitigate methane emission, and to optimize the management of inbreeding. And again, novel methods of sequencing, and again, in particular in nanopore, are also employed in the understanding of the role of epigenetics, thus the effect of the environmental stimuli on diversity and adaptation. So through the study of transgenerational transmission and how, the impact of environment on epigenetic, in particular methylation marks, could influence genetic diversity. And of course, genotyping and sequencing technologies applied to livestock data generate a vast amount of data. In fact, they were really the drivers of the data revolution as well. This rapid growth in data brings significant challenges in storing, processing, and analysis of data in livestock. For data storage, International Public Archives, I report here just a few of them, are really critical for securely storing genomic data and ensuring long-term access. Novel methods are under development for optimal data representation, for optimizing the data representation to improve storage capability. Advanced memory and high performance computer are required to process and analyze large-scale data sets, and machine learning and even more deep learning methods for data analysis and representation are essential for extracting meaningful insight from complex genomic information. Tackling these challenges demand, of course, an approach which is transdisciplinary and combine expertise in genetics, data science, and technologies. While remarkable progress has been made, overcoming this limitation remains crucial and to fully unlocking the potential of genomic data in improving and conserving animal genetic resources. And with that, I'd like to thank you, and I hope I was within the time.